future doctors, let's get right into it. If you're prepping for your micro exams, you absolutely know E. coli is a high-yield topic you can't ignore. So today, we're going to do a super focused, no-fluff breakdown of everything you need to know to absolutely ace that question. Ready? Let's go. So here's the big question, right? How can this one bug cause something as common as a UTI, but also something as devastating as hemolytic uremic syndrome? It's a normal resident in our gut, yet it can be a killer. Understanding this crazy versatility is really the key to mastering E. coli for your exams.
We've laid this out just like you'd build a perfect long answer question. We'll start with the basics, then get into its weapons, the virulence factors, cover what it actually does to the body, and then nail down the diagnosis and treatment. Let's get started.
Okay, first things first. You got to know the family. E. coli belongs to a huge and super important group called Enterobacteriaceae. Think of it as a notorious family. Its relatives include other big names you've definitely heard of, like Salmonella, Shigella, and Klebsiella. Here it is, your perfect, concise definition for the exam. Write this down, memorize it. It's a gram-negative rod, and it's a facultative anaerobe. And don't forget its double life. Most of the time, it's a harmless passenger in our gut, but sometimes it becomes a very serious pathogen.
So everyone in this family shares a few key characteristics, kind of like family traits. They're all gram-negative rods, they all ferment glucose, they're catalase positive and oxidase negative. But, and this is a big but for your lab diagnosis, what makes E. coli stand out? It's a powerful lactose fermenter. That is your #1 clue when you're trying to identify it.
So how does this harmless gut bug go rogue? Well, it's all about the weapons it's carrying. Let's break down its arsenal. These are its virulence factors. E. coli's main weapons are on its surface. These are the antigens we use to identify different strains. You've got the O antigen in the cell wall. The H antigen is for its flagella, which lets it move around. The K antigen is its capsule. Think of it like a stealth cloak that helps it hide from your immune system. And finally, you have the fimbria, or pili, which are like little grappling hooks it uses to latch on and colonize.
Now for the heavy artillery, the toxins. This table is gold. Notice the key differences. ETC makes two types, LT, which jacks up CAMP just like cholera toxin, and ST, which jacks up CGMP. EHC is a whole different beast. Its verotoxin is seriously nasty. It completely shuts down a cell's protein factory, leading to cell death.
Okay, exam hall pressure. It's so easy to mix up LT and ST. Here's a stupidly simple trick that you will never forget. For ST, the stable toxin, just take the S and tilt it on its side. What do you get? It looks like a G, so ST increases CGMP. Easy, right? You're welcome. With an arsenal like that, it's really no surprise that E. coli can cause a massive range of diseases. Let's take a look at its clinical hit list. You know, we always think of E. coli and diarrhea, but get this, it is the #1 cause of UTIs, period. It's also a huge player in neonatal meningitis, hospital-acquired pneumonia, sepsis, and some really nasty wound infections. This incredible range is exactly why it's so clinically important for you to know inside and out. Okay, deep breath. We're now at the part that often messes students up, the 6 different types or pathotypes of E. coli that cause diarrhea. But don't worry, we're going to make this crystal clear. This is your golden ticket for the exam. Seriously. Let's walk through it. EPEC. P is for pediatric diarrhea. Easy. ETEC. T is for traveler's diarrhea and its toxins. EIEC. I is for invasive because it invades your cells. Now, EHEC is the one you really need to focus on. H is for hemorrhagic or bloody diarrhea. Its virotoxin can cause HUS, hemolytic uremic syndrome, which is a life-threatening kidney failure. Super important. And finally, EAEC is aggregative and DAEC is diffuse adherence. The mnemonics tell you exactly what you need to know. All right, so you've got a patient and you suspect an E. coli infection. How are you going to prove it? Let's walk through the lab diagnosis step by step, like a detective. The process is super logical. First, you get the right sample, urine, stool, pus, whatever it is. Then you do a direct smear, you culture it to grow the bug, and finally, you run biochemical tests to get your definitive ID. When you plate it on McConkey agar, you're looking for this classic sign, bright pink colonies. That pink color is the giveaway. It tells you the bacteria is fermenting lactose, which changes the pH and triggers the color indicator. This is your first major visual clue. So what's next? You take one of those beautiful pink colonies, pop it on a slide, and hit it with a gram stain.
And bingo! You see exactly what you expected to see. Gram-negative, pinkish-red rods. This confirms our initial suspicion. And for the grand finale, the final confirmation, we run our biochemical tests. And I've got another great mnemonic for you, ICUT. E. coli is indole positive, but citrate and urease negative. The TSI slant shows an acid-over-acid reaction with gas production. This biochemical profile is the nail in the coffin. It's definitively E. coli. Okay, we've caught our culprit. We know it's E. coli. Now, what do we do for the patient? Let's talk treatment and then do a super quick final revision. Now, this is absolutely critical to remember. For diarrhea caused by E. coli, the most important treatment is not antibiotics. It's fluid replacement. In fact, and listen closely, for EHEC infections, giving certain antibiotics can actually be dangerous. It can cause the bacteria to release a flood of verotoxin, which can push a patient into HUS. So remember, rehydration is king. Antibiotics are used very, very cautiously. Okay, rapid-fire revision time. Let's lock this in. Key ID, gram-negative rod, lactose fermenter, indole positive. ETEC, traveler's diarrhea, LT and ST toxins. EHC, bloody diarrhea, and HUS from verotoxin. Remember that S to G trick for the ST toxin. Lab diagnosis, look for pink colonies on McConkey and confirm with your ICTU tests. And treatment, fluids, fluids, fluids. Be so careful with antibiotics and HUS-causing strains. So we've armed you with everything you need for your exam. But here's something to think about for the future. E. coli is an absolute master of adaptation, and it's getting scarily good at resisting our antibiotics. The rise of multi-drug-resistant strains is already a huge challenge. So what do you think is the next big threat this tiny bacterium is going to throw at us? Something to ponder. Okay, that's it. Good luck with your studies.